Hello everyone, welcome back to my channel. So today I will discuss all about flow cytometry, about this technique. Hello friends, I am Dr. Shaha. I am a molecular biologist with expertise in microbiology, genetics and bioinformatics. On this channel, I break down complex biological concepts into easy to understand content. If you're passionate about science, subscribe and join me on this journey of discovery. Today I will talk about flow cytometry. You can say all about flow cytometry. So what is flow cytometry and what is the use of flow cytometry, how flow cytometry works. So I'll discuss all about these things. So as you know that flow cytometry is a powerful technique used to analyze the physical and chemical properties of cells or particles as they flow through a laser beam in a fluid stream. It allows for the simultaneous measurement of multiple parameters, for example, size, complexity, fluorescence, and more, on thousands of cells per second. So how it works? Simply, the first step is sample preparation, right? So cells or particle that are suspended in a fluid can be used for flow cytometry detection. They are often stained with fluorescently labeled antibodies or dyes that bind to specific proteins or cellular components. After sample preparation, you can say the fluidic system, the next step. So the sample is injected into a stream of sheath fluid which focuses the cells into a narrow single file stream. And after this fluidic system, so the laser and detection steps actually starts. So as each cell passes through the laser beam, it scatters light and may emit fluorescence. So what sort of fluorescence emissions and how the scattering works? So there are actually two types of scatters. Firstly, the forward scatter, which is called FSC, forward scatter cells, that measures cell size. And the second one is side scatter, which is called SSC, side scatter cells. So that measures cell complexity or granularity. And then another things actually are detected, which is very important as well. This is fluorescence. So these measures the presence of specific markers using fluorescent tags. So after the detection, what the next step? So these are the optics and electronics. So the scattered and emitted light is collected by detectors. Detectors could be photomultiplier tubes or PMTs. So then the signals are converted into digital data, which is actually done by these electronics. And after we get the digital data, the next step is data analysis. But this is the most important part of the whole process, because if the data is not analyzed accurately, then the story could be just totally different, So the data is processed using software to generate histograms, dot plots, and other graphical outputs. So the very common software we use for this flow cytometry data analysis is flowjo. And this data analysis allows for identification of cell populations, cell cycle analysis, apoptosis, protein expression, and many other methods as well. So what are the applications of this technique? To be very short, actually, there are multiple applications of this flow cytometry exist. For example, immunophenotyping, like identifying T cells, B cells, cell sorting by facts, like fluorescence activated cell sorting, so how the cells are actually sorted, that can be identified from this flow cytometry technique. Detection of biomarkers, like in cancer diagnosis. So there's a whole lot of immunological markers that can be detected using this technique. Cell cycle analysis. So the cells are in which stage of their cycle, that can be detected by this technique. Apoptosis. Of course, the cell death can be identified from this technique as well. And microbial analysis, for example, some bacterial or yeast populations, how the dynamics works for these can be identified by this method. So what are actually the use of this technique in diagnostic applications? So there are multiple use of these flow cytometry methods in multiple diagnostic purposes at present. For example, hematology and immunology. So we can detect leukemia and lymphoma using this technique. How we can do? Immunophenotyping of blood and bone marrow samples to identify abnormal cell populations. We can differentiate between different types of leukemia, like is it acute or chronic, and lymphoma as well. So there are some common markers that can be identified, and then these diseases can be diagnosed. For example, CD3, CD4, CD8, 19, 20, 34. These all are actually linked with different sort of diseases, and their stages as well. So you can very easily identify these markers and their expression levels in the cells, and probably their locations as well, using this flow cytometry technique. The other important use of this technique, I can say, is for cancer diagnosis and monitoring. So as you know, that there are multiple situations of cancers, multiple stages of cancers that can probably be efficiently diagnosed using this technique. So like the solid tumor analysis. So if we have the solid tumor, and if we can identify the tumor infiltrating lymphocytes, which is called TILs, that can say what sort of tumor it is. And using this analysis technique, we can also detect tumor markers on circulating tumor cells. So if you can identify some of the tumor markers in the cells, then probably you can predict the cancer, or you can say much more concisely what is the stages and what sort of cancer is this. So that's the reason actually we can also use this flow cytometry technique. We can also identify multiple myeloma using this technique. For example, detecting abnormal plasma cells and monitor response to treatment. Uh, we also can identify surface markers like CD38, CD138, and CD56 for this myeloma sort of diagnosis. So the, the use of flow cytometry is not limited to these techniques as well. There are a lot of other purposes. We can also use flow cytometry, for example, to um, understand the systemic lupus erythematosus, SLE disease, rheumatoid arthritis. So we can also measure the factors responsible for these diseases, and we can do this by this flow cytometry technique. Um, we also can use this technique for infectious diseases. For example, HIV/AIDS. So you can monitor CD4 plus and CD8 plus T cells counts, and we can understand the stages or the disease progression. So yeah, we can also track the immune response to antiretroviral therapy using this technique, right? And the diseases which are also associated in immunological responses. For example, different viral and bacterial infections that can cause cell activation or cytokine protection. So we can actually measure these activation states and the related cytokines abundance, so that we can actually say what sort of infection is this and how critical the infection by this technology. So again, these are not actually limited diagnostic purposes for this tool. There's a whole lot of other techniques and currently evaluating the techniques as well for these flow cytometry method. So this is really a powerful method. And I can say that this flow cytometry is very powerful in diagnostics as well. So it has high sensitivity and specificity. It has the rapid analysis procedure. So thousands of cells per second can be analyzed using this technique. There are multi-parametric analysis exist for this tool as well. Like multiple markers can be analyzed simultaneously using this technique. And it's definitely we can also diagnose multiple diseases at a time using this tool. And this technique has the ability to analyze heterogeneous cell populations as well. So to be very specific, flow cytometry is really valuable in oncology, hematology, immunology, and infectious disease diagnostics due to its ability to distinguish subtle differences in cell surface markers and intracellular proteins. So that's it for flow cytometry today. I hope you liked the video, and please subscribe the channel and share this video with your friends so that it can be reached easily. And I invite you for my next video, and until that, stay well. Thank you. See you again.